Homo Sapiens. Who are we? Where do we come from? In Chapter 19, we will continue our look at the beginnings of life on Earth. As we noted at the end of Chapter 18, we will be traveling through the shadowy borderlands that separate the prebiotic world from the biotic world. Somewhere along this shadowy border must lie the secret to Darwin's spark of life. Our focus in this chapter will be on entities classified as non-living entities, namely viruses and viroids. We will be seeking to glean any clues as to how non-living entities made the leap to becoming living organisms. We can be certain from the very fact of our own existence that somewhere along this shadowy border must lie the bridge of life. What lies on the prebiotic side of the bridge of life is veiled in shadows. On the biotic side of the bridge, or very near the bridge, must lie the sin ancestor, or last common ancestor, of all life on Earth. The sin ancestor being the progenitor to the three earthly domains of life, the archaea, the bacteria, and the eukarya. Let's now begin our journey through the shadowy borderlands that separate the prebiotic world from the biotic world in search of the bridge of life. On the prebiotic side of the border, we would find in the far outer margins of the world the organic molecules that serve as the building blocks for the evolution of life. As we draw closer to the border, there appear molecules of increasing complexity, such as RNA and other macromolecules, acting as stepping stones along the path to self-replicating entities, shadows of a hypothetical RNA world. Even closer to the border yet, we would find entities classified as non-living entities, namely viruses and viroids. Viruses and viroids seem to mysteriously straddle the border between the prebiotic and biotic worlds, the ghostly foot in each world. Let's begin our look at viruses and viroids with some basics. We will tackle the viruses first. To begin with, what exactly is a virus? A virus is defined as a small infectious agent that can only reproduce by hijacking the reproductive machinery of a living cell. Most viruses range in size from 20 to 300 nanometers, a nanometer being 1/1000th of a meter. As a comparison, an E. coli bacterium is around 2,000 nanometers in length. A red blood cell is about 8,000 nanometers in diameter. A virus or single viral particle existing outside a living cell is called a virion. A virion is typically composed of a nucleic acid, DNA or RNA, surrounded by a protective coat of protein called a capsid. Some viruses display a third component, an envelope of lipids that encases the protein coat when the virus is outside a cell. The first scientific article describing a non-bacterial or viral agent was published in 1892 by Russian biologist Dmitry Ivanovsky when investigating a non-bacterial pathogen infecting tobacco plants. In 1898, Martinus Beijerinck, a Dutch microbiologist, conducted experiments that led him to the discovery of the tobacco mosaic virus. Beijerinck published a paper describing his experiments in attempting to isolate the tobacco plant pathogen through filtration. In his paper, Beijerinck termed the pathogen a virus to indicate its non-bacterial nature, the word virus deriving from the Latin meaning a poison or potent juice. Beijerinck proposed in his paper that the virus was some kind of contagium vivum fluidum, or contagious living fluid. The actual physical structure of the virus wasn't understood until the invention of the electron microscope in 1931, and further research carried out between 1935 and 1941 revealed the virus to be a particle. Viruses are thought to be the most abundant biological entities on the Earth, with estimates in the range of 10 to the 31st power viral particles inhabiting the Earth's biosphere. Science so far has described about 5,000 virus species in detail, though millions of types of viruses exist. All three domains of life on Earth, the archaea, bacteria, and eukarya, are subject to viral infection. So, if the most primitive forms of life on Earth today are subject to viral infection, does it follow that viruses were around prior to or at the beginnings of life? This question presents a bit of a conundrum in that the very definition of a virus indicates that it can only reproduce via a living cell. So, how could viruses have reproduced and survived prior to the appearance of a living cell when the virus itself is dependent on same said living cell for its reproduction? All of this head-scratching brings us to the question of the origin of viruses. Scientists have developed three basic hypotheses for the origin of viruses. The first hypothesis is called the regressive hypothesis. The regressive hypothesis postulates that viruses may have once been small cells that preyed upon larger cells. Over time, the smaller parasitic cells lost genes not required for survival, causing them to devolve down to a viral level. The second hypothesis is the cellular origin hypothesis. The cellular origin hypothesis states that the viruses may have evolved from bits of DNA or RNA that escaped from the nucleic acid of a larger cell. This escaped nucleic acid could have come from plasmids, sections of naked DNA that can move between cells. Another hypothetical escapee could be transposons, which are molecules of DNA that replicate and move around to different positions within the genes of a cell. The third hypothesis is the coevolution hypothesis, or the virus first hypothesis. The coevolution hypothesis proposes that as the first cells were beginning to evolve in that chemical milieu of complex organic molecules on the early Earth, some form of viral entity may have been an evolving chemical constituent of that same complex organic soup. These viral entities thus evolved in tandem with the first living cells possibly existing in some inchoate chemical state prior to the appearance of the first living cells. Both viruses and living cells depend on DNA or RNA as information storage systems for reproduction. Whereas living cells have evolved a cellular membrane and a metabolic system, along with the core information handling system to maintain homeostasis for survival, viruses simply evolved a maintainable information handling system, depending upon the machinery of living cells for their viral reproduction. Since all of life is basically chemistry anyway, it's possible that viruses are just the result of co-evolving complex chemical molecules. And this brings us to viroids, which may help to provide further insight into the origin of viruses, as well as further insight into the origin of life itself. What exactly are viroids, and how do they differ from viruses? Let's start with some basic facts to familiarize ourselves with viroids. Viroids are the first known representatives of a new domain of subviral pathogens. Viroids, which are plant pathogens, were first discovered and named by Theodor Otto Diener in 1971. Diener, a plant pathologist working for the U.S. Department of Agriculture, discovered viroids while investigating the causes of spindle tuber disease in potato plants. The viroid discovered by Diener is called the potato spindle tuber viroid, often abbreviated to PSTVD. Viroids are described as naked, circular, single-stranded RNA molecules that do not code for protein, yet replicate autonomously when introduced into a host plant. The naked RNA molecule of the viroid, naked meaning the RNA molecule is not protected by a surrounding structure, is one of the biggest differences between viroids and viruses. Viruses having their nucleic acid, DNA or RNA, encapsulated by a protective capsid composed of protein. The 30 known varieties of viroids are classified into two families, postbiviroidy and avsunviroidy. The distinction between the two families being that postbiviroidy replicate in the host plant cell nucleus, whereas avsunviroidy replicate in the host plant cell chloroplast. Viroids are intriguing on a number of levels. First of all, the origin of viroids is cloaked in mystery. In 1989, Theodore Diener, who discovered the viroids, set forth the hypothesis that viroids represent a relic of a hypothetical RNA world. Before we dive into more details about the RNA world implications of viroids, let's take a minute to look at the physical structure of a viroid. This will give us a better understanding of some of the basic aspects of viroids, as well as RNA genomes in general. We will use the potato spindle tuber viroid as our example in this instance, henceforth referred to as the potato viroid. The potato viroid has a genome consisting of 359 nucleotides, forming a circular single-stranded RNA macromolecule. Research indicates that the potato viroid, in addition to its circular form, also displays a secondary rod-like form. This secondary form results from the folding of the circular RNA into a rod-like structure. Research has revealed that this rod-like form of the viroid facilitates the viroid's entry into the RNA trafficking system of the host plant, serving as a mechanism for infection of the host plant by the viroid. In the case of the potato viroid, the rod-like form is composed of 27 loops or bulges flanked by double-stranded helical sections, as we can see in this 3D model. Research seems to indicate that these loops play a critical role in mediating systemic trafficking across cell boundaries, as well as playing a role in viroid replication. Many types of cellular RNA, both in plants and animals, are involved in a system of intercellular trafficking or cell-to-cell movement for the purpose of intercellular communication and regulation of various biochemical processes necessary for homeostasis in living organisms. As an example, microRNAs trafficking from the budding stems down to the roots of the Arabidopsis thaliana, a small blooming plant commonly called a mouse ear cress, are thought to play a role in the regulation of a gene involved in phosphate homeostasis. If we take a look at this simplified model of a group of plant cells, we can note the interconnecting tube-like structures. These tube-like structures are called plasmodesmata, which are microscopic channels that transverse the cell walls, creating an intercellular highway system used to transport from cell to cell vital biochemicals and genetic resources necessary for the plant's survival. Viroids in some ways can be viewed as interlopers, joyriding on a living plant's existing system of intercellular RNA highways for the purpose of facilitating their own viroidal self-replication. If we take a moment to examine this 3D structural representation of the rod-like secondary form, we can see the 27 loops with the flanking helical sections. The entire length of this rod-like structure is approximately 37 nanometers long. By comparison, a human hair on the average may be around 68,000 nanometers in diameter. The 359 nucleotides forming the potato viroid genome are built from the nucleobases guanine, cytosine, adenine, and uracil. Between the 27 loops, we can see the tighter helical sections, which are the result of the hydrogen bonding of the nucleobases. All of these seemingly simple yet intricate biochemical structures present in the viroid play a role in impacting a living organism, the host plant, on a system-wide basis. It boggles the mind to think that so much of life is make or break on such a minute biochemical level. The viruses that have plagued humankind across the ages, from smallpox to Ebola to influenza, function on this same minute biochemical level, yet have an impact on a planet-wide scale. These smallest of entities have no doubt played a role in shaping the evolution of life on our planet. Getting back on topic, Diener noted that viroids display a set of properties which make them good candidates for survivors of an RNA world that is hypothesized to have existed prior to the evolution of cells. The first telltale property is the viroid's small RNA-based genome size. The viroid genomes are extremely small, ranging from 246 to 467 nucleotides. By comparison, the genome of one of the smallest known viruses, the porcine circovirus, is a single loop of DNA which is 1,768 base pairs long. As a further comparison, the human genome consists of over 3 billion DNA base pairs. The advantage of a small genome size is that it helps to avoid error catastrophe caused by error-prone replication. Error catastrophe being the extinction of an organism through excessive undesirable mutations. Viroids also have a high guanine and cytosine content, which increases nucleic acid thermodynamic stability as well as enhancing replication fidelity, important survival characteristics for a viroid. As we noted earlier, guanine and cytosine are two of the 4 nucleobases that comprise the RNA genome in viroids, the other 2 nucleobases being adenine and uracil. Another property that appears to be a relic of the RNA world is the viroid's circular RNA-based structure, which obviates the need for mechanisms such as genomic tags that are found at the ends of linear genomes to prevent information loss. Viroids also display structural periodicity, allowing for modular assembly, thus facilitating the building of more complex genomes. Structural periodicity being the use of small repeating structures to build larger, more complex structures, a simplistic example of this concept being Legos. The RNA-based viroids also lack a protein coding ability that is consistent with a ribosome-free habitat, which is another indicator of a precellular RNA world. Ribosomes being the site of protein synthesis found in all living cells. Some viroids display a ribozyme-mediated replication, ribozymes being a fingerprint of the RNA world. To explain, ribozymes, or ribonucleic acid enzymes, are RNA molecules that are capable of catalyzing specific biochemical reactions. The discovery of ribozymes in 1982 demonstrated that RNA can function both genetically and catalytically, a dual role which contributed to the hypothesis that RNA may have played an important role in the evolution of prebiotic self-replicating systems. Present-day viroids, however, are no longer self-replicating. They may have lost that functionality when they became parasites of plants. Unlike viruses, which are parasites of host translation machinery, viroids are parasites of cellular transcription proteins and depend on cellular RNA polymerase for replication. RNA polymerase, or ribonucleic acid polymerase, is an enzyme that produces primary transcript RNA, which is a single-stranded RNA product synthesized from information stored in the DNA of a living cell. Primary transcript RNA is in turn processed into messenger RNA, transfer RNA, and ribosomal RNA, which are critical to carrying out cellular processes. RNA polymerase is found in all living organisms as well as a large number of viruses and is essential to life. Both viroids and viruses can essentially be seen as hackers of the DNA-RNA reproductive machinery of living cells, hijacking the cellular mechanisms for their own viral reproduction. One intriguing aspect of viroids is that they may represent a step, or at least some insight into a step, in the abiogenic evolution of non-living entities into living organisms. To illuminate this last statement, let's take a look at an unusual virus that has viroid-like characteristics. The hepatitis delta or hepatitis D virus is a small spherical virus about 36 nanometers in diameter. Structurally, the hepatitis D virus has an outer envelope composed of 3 types of protein as well as host lipids, with an inner nucleocapsid which contains a single-stranded circular RNA consisting of 1,679 nucleotides. Hepatitis D is the smallest known virus to infect animals and is the only animal virus with a single-stranded circular RNA. It is hypothesized that hepatitis D may have originated from viroids. If hepatitis D did evolve out of a viroid population, then it may speak to the very process of evolution at the smallest of levels, where complex macromolecules, pushed and pulled by the forces of chemical interaction, vibrate in an unseen chaotic dance that ripples up through all of the levels of the Earth's biosphere, molding and shaping all the forms of life that we can bear witness to across the eons of our Earth's existence. If physics reveals to us the nature of our universe, and chemistry reveals to us the nature of life. All that grandiose verbiage aside, we are still looking for Darwin's spark of life. All signs point to it being some unique interaction of physics and chemistry. In Chapter 20, we will continue our search for who we are and where we came from as we continue our quest for the beginnings of life.